thank you, thank you. Following Jean-Luc's wonderful presentation on chemistry and phytochemistry, I will do my best, okay? So my name is Valerie Schini-Kerth from Strasbourg University, and we are interested in natural products and the health effects. And why are we so interested? Because cardiovascular disease, as you know, is still the major leading cause of death. It's increasing globally. You can see that the number of... oh, is it working? Yes.
This one is over, I see.
Yeah, that would be good. Okay, as you can see on the left-hand side, the number of deaths is still on rising lately, as well as the prevalence and the years lived with disability. So there's still a big challenge how to protect our cardiovascular system. And this increase in cardiovascular disease is mostly linked to the aging of our population. You can see from the 1950 to the estimated what's happening in 2050, we have more and more people older than 80 years of age. And of course, age is an increase, cardiovascular risk. You can see on the green light, with increasing age, there is a lifetime risk associated. And this is further increased if you have one risk factor which is not optimal, even further with one elevated risk factor, even with one major risk factor. And of course, if you accumulate risk factors, you can see that cardiovascular events may appear even much earlier. And those cardiovascular risks are mostly linked to, if I can see, yes, those cardiovascular risks, you can see here on the right-hand side. Of course, smoking is a major cardiovascular risk, increased body mass index, physical inactivity, an inappropriate diet, and of course, metabolic diseases like increased levels of cholesterol, LDL cholesterol, increased systolic blood pressure, or diabetes. And the risk associated with cardiovascular disease is mostly the origin is ischemic heart disease and stroke. You can see that ischemic heart disease accounts for about more than 49% of deaths, followed by ischemic stroke. Now, what is the underlying mechanism of those ischemic heart disease? First of all, which are the areas they are affecting? Mostly the heart circulation, the coronary circulation, leading coronary heart disease, heart failure, but also brain circulation with stroke and peripheral artery disease. Those are the blood vessels which are at risk. And the underlying mechanism is mostly the development of arteriothrombotic events. This actually starts very early on in your life. You can see, starting from the fourth decade, we can see appearance of early lesion of atherosclerosis. Of course, no clinical impact, which will further progress and become an atherosclerotic plaque. And this plaque the problem can be, there can be a lesion or a rupture which leads to an atherothrombotic event, and of course, if it happens in the coronary circulation, can lead to myocardial infarction. And when we look carefully at the development of these atherosclerotic plaques, we can see that very early on, what we can see as an alteration is actually the appearance of endothelial dysfunction. This is one of the early events we can detect even at the stage of atherosclerotic lesion. And this endothelial dysfunction is linked to an impaired formation or an increased degradation of nitric oxide, the major vasoprotective factor, increased oxidative stress, monocyte adhesion, lipid accumulation. So then, of course, we need, if there are approaches to protect the endothelial dysfunction, the initiation and development, this would be an interesting approach to prevent the development of cardiovascular disease. Now, lately, it has been published here the top risk factor related to overall disease burden, and you can see the top one is actually the dietary risk. An unhealthy diet is by far the most important risk factor, followed by tobacco smoking, high blood pressure, body mass index, physical inactivity, and then the diabetes and hypercholesterolemia. And very recently, the American Heart Association, but also the European Society of Cardiology, has very important messages for the public regarding health. You can see that the cardiovascular burden is attributable to modifiable risk factors. Here we have the 8 modifiable risk factors indicated: the diet, physical activity, blood glucose level, increased weight, hypercholesterolemia, high blood pressure, smoking, and a relaxing sleep. And when we look carefully on the right-hand side, well, you can see the prevalence estimates of poor, intermediate, and ideal cardiovascular health. You can see that among the 7 modified cardiovascular risk factors, the one which comes out the worst is the diet, a healthy diet. And you can see that those are data from the USA. Regarding a healthy diet, we have less than 0.2% of the population has a healthy diet. Less than 20% have an intermediate diet, and more than 80% have a very poor diet. Now, how can such a diet impact the cardiovascular system? And what can we do? You can see here there is now more and more evidence indicating that actually our diet is very important for protecting our cardiovascular system. Here's published the first primary prevention study regarding the impact of Mediterranean diet on the cardiovascular system. They enrolled 7,447 persons. They were not persons with a high cardiovascular risk who were free of cardiovascular disease when they were included in the study. And they were either on a controlled diet, there was people from Spain on a controlled diet, or a Mediterranean type diet with daily intake of nuts or extra virgin olive oil. And you can see over the 5 years of the study, there was a very huge benefit with a relative risk reduction for approximately 30%. This was a primary prevention study. And here they also very recently on the same topic published in a secondary prevention study. You can compare the Mediterranean diet to a low-fat diet in preventing major cardiovascular events. It was the CardioPrev study in 1,002 patients with established coronary heart disease. And the Mediterranean diet was compared to a low-fat diet, and again, the Mediterranean diet did much better with a 26% reduction in the risk, in the relative risk. We also have some interesting data here from a study from China, where they looked at the intake of red food consumption and major cardiovascular disease in China. It's a very big population, more than 500 adults, and they look the development of cardiovascular death or major coronary events with portions of intake of fresh food. And you can see a negative relationship. The more food intake the people have, the reduced risk they have for cardiovascular death and major events. And what is interesting, 30 portions per month, which is 1 portion of food per day, was actually associated, you can see here, with 37% reduction in the risk of cardiovascular death and a 30% risk reduction in major coronary events. So really very strong effect. Now, how can we explain this impact of the diet on the cardiovascular system? And we know since Professor Fürschger's discovery of EDRF, which is nitric oxide produced from endothelial anosyntase, the major vasoprotective factor in the cardiovascular system, which can be modulated by a lot of different local factors, but also the most important one is shear stress activating endothelial cells to produce nitric oxide in a continuous manner over 24 hours, and this NO is important for vascular production, but also a strong inhibitor of platelet and monocyte adhesion and activation. And we know now since more than 20 years, when the studies have started, that actually there are nutrition-derived activators. We can exactly do the same thing, activate endothelial cells to produce nitric oxide. We have cocoa polyphenols, red wine polyphenols, berries, and green tea polyphenols. I would like to share with you an overview of some of the studies we contributed. And you can see here very early on in 2000, we studied the red wine polyphenol extract. Only the polyphenols were isolated from red wine from the French Ministry of Agriculture. And the red wine polyphenols in porcine coronary arteries contracted either with endothelium intact artery caused full relaxation We remove the endothelium, absolutely no effect anymore. So we have a strict endothelium-dependent relaxation. And what is interesting to see, here we have the concentration response curve. You can see 10 microgram per milliliter of the extract. We have nearly 100% relaxation. And just to indicate you that 3 microgram per milliliter of polyphenols have been detected in plasma of a subject after drinking 1 glass of red wine. When we look at the mechanism underlying this relaxation, we could see that nitroargine, the inhibitor of endothelial anosynthase, significantly reduced the relaxation. It was also inhibited by the 2 toxins here, which inhibit the endothelium-dependent hyperpolarization component, and the combination of 2 was associated with a full inhibition of the relaxation, indicating that this polyphenol, when polyphenol-induced relaxation, is mediated by nitric oxide and endothelium-dependent hyperpolarization. As a pharmacologist, of course, we do not want to motivate people to drink red wine. We have, of course, the ethanol component, which has a deleterious impact. So we were looking for different sources of polyphenols being able to have a beneficial impact on the cardiovascular system. And for this, we worked with the food industry. We were able to get here 13 different food, either as a juice or as a puree, which are used by the food industry in Europe, and we tested the activity of those compounds in the coronary artery system to do a kind of screening. Here in each graph you have in red, it's the same curve, which is the Concord grape juice, which is a potent activator of the endothelial formation of nitric oxide, and we were able to identify 5 products here which are more active than the grape juice. Here we have 3 products less active, and here we have products with very little activity. And this was very surprising for us because all the products have a high level of polyphenols, more than 1.7 gram per liter. So it's not, if you have a product with a high level of polyphenols, that you will have biological activity. You really need to identify what are the compounds, and as Jean-Luc said, we need new techniques to identify which structure is responsible for this activity. And again, the ones with the highest activity, you can see here on the left, is the aronia berry, the blackcurrant berry, blueberries, cranberry, and lingonberry. Those products are products really of major interest for the cardiovascular system. And we also looked, based on our studies, what is actually on the shelf of the supermarket. What kind of products do we buy as consumers? And here we compare 6 different fruit juices, which are blackcurrant juices, which are you can buy in the supermarket in France and on a neighbor city in Kiel, Germany, just crossing the Rhine River. And you can see actually that what we observed is actually the biological activity to induce endothelium-dependent relaxation was actually quite heterogeneous. Some food juices had very strong activity, and some had a very poor activity. And this is, first of all, important because it means that as a consumer, what we buy, we have no idea about the intrinsic quality of those juices. And with Joël Pansmail and our colleagues from the University of Liège, we looked at the antioxidant activity of those juices and the biological activity to see whether or not the antioxidant properties is of importance. And actually what we were able to see is actually the antioxidant activity is not associated with the biological activity. However, when we analyze the anthocyanin content, which gives the color to the blackcurrant juice, you can see that the total anthocyanin and also the individual anthocyanins were highly correlated to the activity, indicating that anthocyanins are involved in developing the biological activity of the blackcurrant juice. We also were interested to identify, as pharmacologists, what are the underlying mechanisms leading to endothelial cell activation. And here we stumbled in some surprising data at the first time. You can see here on the left-hand side, so here we do the experiments in presence of indomethacin and caryoptotoxin and apamine, just to focus on the nitric oxide-mediated relaxation. So this relaxation to Concord grape juice is entirely due to nitric oxide. And you can see when we based on the experiments, we tested whether or not there was a pro-oxidant response in endothelial cells, triggering activation of endothelial anosynthase. And you can see that the SOD membrane permanent analog, MNTMPYP, had a very strong inhibitory effect, whereas extracellular SOD, which is not able to cross membranes, was inactive. We also looked whether or not hydrogen peroxide was involved, and you can see that PEG catalase, able to cross membranes, also very strongly inhibited relaxation, whereas extracellular catalase was inactive, indicating that somehow we have an intracellular pro-oxidant response in endothelial cells, triggering the cascade leading to the activation of endothelial endosynthes. And this cascade was actually involving the activation of SARC. You can see the PP2 inhibitor significantly inhibited, and also PI3 kinase was inhibited. Workmannin, a classic inhibitor, is inhibiting the relaxation significantly. And indeed, when we look using Western blot analysis on cultured coronary artery endothelial cells now, when we expose to the Concord grape juice, you can see within minutes here, you can see an increased phosphorylation first of SARC, which will trigger the phosphorylation of AKT, and AKT is well known to activate endothelial endosynthes by phosphorylating serine 1177, which is an activator site. And you can see very strong phosphorylation up to 30 minutes. And this phosphorylation, this activation of this cascade, SARC, AKT, eNOS, in response to Concord grape juice, is highly sensitive to inhibitors of oxidative stress. Here you see PEG catalase, PEG SOD, and MNTMPYP, the membrane permanent analog of SOD, indicating that superoxide ions and hydrogen peroxide are involved in the activation of this cascade. And we were of course, interested. If you want to protect the cardiovascular system, it's not a bolus of NO which will protect in the long term. You need a little bit more NO formation, like with shear stress, in a sustained manner. So we looked here, activation of endothelial cells. This is the Aronia juice, and you can see up to 24 hours we looked here. And you can see phosphorylation of AKT is maximum 2 hours, then decreases slightly. But phosphorylation of eNOS is observed for 24 hours. It's a very long-term event. And again, we still do not know exactly how can we explain such a sustained effect. And when we looked at the control level of eNOS protein level, the non-phosphorylated one, what we were able to see was actually a time-dependent increase in the level of eNOS protein. And that's why we were also interested to see whether or not natural products may stimulate the expression of, upregulate expression of eNOS. And you can see on the right-hand side, I should not forget, bradykinin is a physiological agonist, and you can see that the phosphorylation of eNOS is very transient. After 30 minutes, there is absolutely no activation of eNOS anymore. Whereas with a polyphenol, an active polyphenol product, you can see, we see up to 24 hours activation of endothelial endosynthes. Very interesting. And so we went further to assess what is the effect of polyphenols on expression of endothelial endosynthes. And you can see data with purple grape juice. You can see with increase in time, there is an increased level of eNOS mRNA. We have an increased protein level here at 8 hours, at 24 hours, and this is associated with an increased formation of nitric oxide here, indicated after 24 hours. So indeed, we have a long-term effect, not only short-term effect stimulating NO formation, but also a sustained effect associated with an upregulation of eNOS and continuous formation of nitric oxide. This is exactly what we are looking for to protect the cardiovascular system. And when we looked at what is the underlying mechanism accounting for the upregulation of eNOS expression, you can see here the increased eNOS mRNA level in response to Concord grape juice is also significantly reduced by using the SOD and catalase membrane permanent analogs. And we could demonstrate that indeed Concord grape juice increases the formation of ROS, here assessed by dihydroetidium, and this is prevented by the PEG catalase and the SOD mimetic. And you can see here we looked about more precisely what is the mechanism involved in the upregulation of eNOS expression, and we looked at the FoxO transcription factor, which are actually negative regulators of eNOS expression. And we could see that the Concord grape juice causes phosphorylation of the two FoxO transcription factors. This is a redox-sensitive event. And when FoxO 1 and 3 are are phosphorylated, they are no longer associated with the enos promoter, which they can no longer inhibit enos expression, which of course will contribute to enos expression. So based on this first part of the study, we have now identified that anthocyanin-rich products are of interest to protect the cardiovascular system. And they activate endothelial cells via a pro-oxidant mechanism in endothelial cells, leading to the activation of the PI3 kinase AKT pathway, phosphorylating enos, stimulating enos production formation in a sustained manner. And also, long-term effects leads to upregulation of enos expression and more enos protein, of course, sustained formation of nitric oxide, which means that they are interesting products to protect the cardiovascular system. Now, when we look carefully in the cardiovascular system, atherosclerosis will develop not everywhere in the cardiovascular system. It develops in the coronary circulation, in the carotid bifurcation, femoral bifurcation, at the renal level. And when you look carefully in the coronary circulation here in the schematic, you can see in black, this is where actually the development of atherosclerotic plaques is happening. And you can see those are bifurcations in the main coronary arteries. Now, why are those bifurcations at such a high, at the target? And this can be explained to the fact that where you have bifurcations or curvature, you have disturbed flow, low levels of shear stress, which means endothelial cells are not activated in an appropriate manner. That means they will not produce enough nitric oxide to protect the arterial wall. And you can see this compared to, for example, the aorta, which is, of course, a tube, and where you have a steady laminar flow, high levels of shear stress, high levels of NO formation. And you can see this very easily when you look what's happening here in the aortic arch. You can see at the inner curvature, you can see that there is expression of vascular cell adhesion molecule. This is an activator of endothelial cells, whereas at the outer part of the ascending arch, you can see absolutely no expression of VCAM. And when we look very carefully here, this is the aortic arch, which is open. You can see that the inner curvature presents senescence. Senescence-associated beta-galactosidase, the blue staining, is in the inner part of the curvature, but the outer part is protected. And this is due to the fact that we have a deficit in NO formation. Less NO in the inner curvature leads to activation of endothelial cells, expression, for example, of VCAM. Now, senescence is a very important event because there are now more and more data indicating that actually premature senescence of endothelial cells is associated with several cardiovascular risk factors. It is induced in endothelial cells by oxidative stress, high levels, but also by activators involved in cardiovascular risk, like angiotensin II, oxidized LDL, high glucose, inflammatory cytokines. And this causes a major change in the biochemical activity of endothelial cells. They show senescence-associated beta-galactosidase activity, expression of markers of senescence, inhibitors of cell cycle like P16, P53, P21, excessive formation of ROS, but not a low level, a high level of ROS, and of course, enos will be downregulated, NO will be decreased, leading to endothelial dysfunction. Now, there are very interesting data published in 2008 in Circulation Research indicating that the induction of senescence leads to endothelial cell dysfunction. It's a very early event. And what this group did, they actually overexpressed the P53, which is a marker of senescence inhibiting cell cycle, only in endothelial cells. You can see here beautiful endothelial cells, which is expressing P53. The control vessel has no overexpression of P53. And what is the consequence at the functional level? You can see that expression of P53, specifically in endothelium, is associated with a significant impairment of the endothelium-dependent relaxation. And also, the formation of nitric oxide is significantly decreased, indicating that senescence leads into endothelial dysfunction. And this is why we were interested to see what is the effect of active polyphenols on endothelial senescence. For this, we prepared a blackcurrant extract so that we work only on polyphenols. We're using chromatographic column. And what was interesting to see, and that's why we shifted to prepare blackcurrant extract, because in blackcurrant juice, we have anthocyanins, but there are 4 major ones. And the 4 major ones are mostly conjugates of delphinidine to glucose or rutinoside to different sugars, and also cyanidine conjugated either to glucose or to rutinoside. And we were able to test the different compounds. First, to show you that the blackcurrant extract causes endothelium-dependent relaxation. It's really a strict endothelium-dependent relaxation. You get rid of the endothelium, absolutely no effect. And again, highly effective. And you can see here on the right-hand side the formation of nitric oxide assessed by this fluorescent probe, DAF-FN. You can see that the formation of NO is actually very similar to what we see with bradykinin, the physiological agonist. And this is NO, it is inhibited by nitroarginine, as expected. Now, we tested the effect of this blackcurrant extract on endothelial senescence in 2 different models. On the left-hand side is a replicative senescence model when you make passages of endothelial cells in our coronary artery endothelial cells very rapidly at P3. We already have 40 to 50% of endothelial cells in a senescent state. And you can see that this replicative senescence is inhibited in a concentration-dependent manner by the blackcurrant extract, here 3 to 10 microgram per ml, so you need low concentration. And on the right-hand side, we also developed a premature model of senescence. We exposed endothelial cells to angiotensin II for 24 hours, and you can see S-beta-gal activity significantly increased. And here also, pre-treatment with blackcurrant extract was able to inhibit in a concentration-dependent manner the induction of senescence in response to angiotensin II. We also were able to confirm the induction of senescence looking at the small proteins, the senescence markers, which are inhibitors of the cell cycle. You can see that the angiotensin II induced upregulation of P53, P21, and P16 are all very nicely, strongly prevented by the blackcurrant extract. Now, what is the consequence if we prevent induction of senescence? This was tested here. We looked at the formation of nitric oxide using the fluorescent probe. In response here to bradykinin, we have an increased formation of nitric oxide. If now we pre-treat the cells, endothelial cells, with angiotensin II to induce premature senescence, you can see that the addition of bradykinin at the end had almost no effect. Those cells were no more able to produce nitric oxide. However, if we pre-treat the cells with the blackcurrant extract before applying angiotensin II, bradykinin is still able to stimulate nitric oxide formation. So those compounds are able to prevent the induction of endothelial senescence. Now, what is the mechanism? How do those anthocyanins interact with endothelial cells? This is a key and important question. And we stumbled in 2014 in this paper published by a group from China, where they actually look at delphinidine, which is delphinidine-3-O-glucoside. They looked at the protective effect of delphinidine-3-O-glucoside on oxidized LDL-induced apoptosis. You can see very strong induction of apoptosis, which was inhibited in a concentration-dependent manner by delphinidine-3-O-glucoside. And actually what they were able to show is that they were able to see uptake of anthocyanins using autofluorescence. In UVEX, you can see the red color is the autofluorescence of anthocyanins appearing in endothelial cells, and also in the isolated thoracic aorta, when it was exposed to delphinidine-3-O-glucoside, they were able to see uptake, the red color here, in the endothelium, in endothelial cells preferentially. And when they looked what is the mechanism, they were able to show that actually D-glucose was inhibiting the fluorescent signal, the accumulation of delphinidine-3-O-glucoside. It was also inhibited by fluorescein. This is a non-selective inhibitor of sodium-glucose cotransporters. And when they down-regulated expression of SGLT1 by using an siRNA, they were able to significantly decrease the accumulation of delphinidine-3-O-glucoside, indicate that SGLT1 was important for the uptake of the delphinine-free glucoside. And we followed on this pathway using our blackcurrant extract. And we looked phosphorylation of enos in response to the blackcurrant extract, and we were able to show that actually a highly selective SGLT2 inhibitor, empagliflozin, had no effect. However, the dual inhibitor SGLT1, SGLT2, sotagliflozin, was able to fully inhibit phosphorylation of enos by the blackcurrant extract. And the less selective SGLT2 inhibitors, dapagliflozin and canagliflozin, also had an inhibitor effect. And you can see also on the right-hand side on the formation of nitric oxide induced by the blackcurrant extract, the highly selective SGLT2 inhibitor had no effect, whereas the dual SGLT1, SGLT2 inhibitor and the less selective SGLT2 inhibitor prevented NO formation, indicating that the sodium glucose cotransport, in particular SGLT1, is important for the uptake of conjugated anthocyanin into endothelial cells. And we further got evidence that indeed the uptake of blackcurrant extract in endothelial cells was decreased in a concentration-dependent manner by increasing concentration of D-glucose. Mannitol was inactive, so it's a D-glucose competitive event. And we can see on the right-hand side, decreasing D-glucose or D-glucose and sodium also decreased the accumulation of blackcurrant anthocyanins in endothelial cells. And then, of course, the question we asked, do endothelial cells express SGLT1 and SGLT2? We want to have direct evidence, because SGLT2 inhibitors are well known to protect the cardiovascular system. Lately, we have major clinical trials indicating protective effect in heart failure with preserved ejection fraction, and also without preserved ejection fraction. And we looked at SGLT1 and 2 expression on the T cells using immunofluorescence staining. We were able to have a clear signal. And when we looked at the protein level, we were able to detect SGLT1 in the T cells. Here, a positive control kidney and intestine. And when we looked at the SGLT2 expression, we were able to see SGLT2 even in the T cells, which was completely unexpected, because it was, in the literature, it's the kidney, the major organ where you see SGLT2 expression, but on the T cells also express SGLT2. And then we were interested, of course, to see, are all conjugated anthocyanins active in our model? And here we tested cyanidin-free glucoside, beautiful uptake. We boost the autofluorescence using Naturstoff reagent A. And you can see the delphinine-free glucoside also showed very nice uptake. The rutinoside conjugated cyanidin and rutinoside, active, very poor uptake. So the uptake is dependent on the glucose, the type of sugar. Glucose is associated with a strong uptake. And this uptake, you can see, is inhibited by the dual SGLT1 and SGLT2 inhibitor, sotagliflozin. And here are the cumulative data. Now, senescent on the T cells is our target. So we looked what's going on in senescent on the T cells. And here you can see expression of SGLT1 in healthy on the T cells, in senescent on the T cells. And upregulation of SGLT1 expression in the pathologic on the T cells. And here you see the data for SGLT2, low levels in the healthy on the T cells, but a nice upregulation in the pathologic, in the senescent on the T cells. And that's why we looked also at the uptake of blackcurrant extract here compared in P1 and P3, healthy, senescent on the T cells. And you can see the uptake in the healthy on the T cells was even further increased. It was occurs in a greater extent in the senescent on the T cells. And you can see the sotagliflozin, the dual inhibitor SGLT1 and 2, had a significant inhibitor effect in P1 cells, but look what's happening in the senescent on the T cells, very strong inhibition. And we looked also in what's the effect of the SGLT2 inhibitor, no effect of the uptake in P1, the healthy cells, but significant inhibition in SGLT2. And that's why we also looked what's happening in vivo. And we looked at SGLT1 and 2 expression actually in the aortic cross. You can see VCAM is upregulated because we have a site at risk. And you can see upregulation of SGLT1, upregulation of SGLT2, which then brings us to this conclusion that actually glucose-conjugated anthocyanins of cyanidin and delpidine appear extremely interesting to target endothelial cells which are characterized by premature senescence. And of course, we need further data to confirm that. I have a few more minutes? No. I will skip. We did an in vivo study in old animals regarding the effect of anthocyanins. I will show you very briefly. The old animals had hypertension, almost a high level of systolic blood pressure, which was significantly increased by the intake of blackcurrant juice. We were able to show that the blackcurrant juice intake is decreasing the hypercontractility of the arterial wall, reducing oxidative stress in the arterial wall in the old animal. And this is associated with a very nice improvement of the endothelial function. The overexpression of eNOS is reduced, the expression of P53, senescence marker, and activation of endothelial cells like by MCP1. Also, the local angiotensin system is decreased. In the old animal, you see ACE is overexpressed, AT1 receptor is overexpressed. Intake of blackcurrant juice has a very nice protective effect. So in conclusion, you can see that finally anthocyanins appear as interesting products. Again, we now have an idea that it's not all structure of anthocyanins. It depends on what kind of sugar is conjugated. And again, this overexpression of SGLT1 and 2 at arterial sites at risk makes it very attractive that we can target endothelial cells, the pathologic one, where we want to protect the cardiovascular system. And finally, to conclude, I would like to really acknowledge all the PhD students and the postdoc who did those beautiful studies, and our senior scientists in our international collaborations. And here's our new Biomedical Research Center at Strasbourg, of Strasbourg University. Thank you very much for your attention.
Okay, time for one short question.
Yes,
Sorry.
I'm up here. Yes, thank you very much for this presentation. My name is Roland Seifert. I'm the editor-in-chief of the Naunin Schmiedeberg's Archives of Pharmacology. And we got many, many hundreds of submissions on phytopharmacology each year. And I would like to take this opportunity to remind the pharmacological community, if you submit papers, make sure that in addition to an extract, you also include studies with purified compounds to elucidate the mechanism, as we have seen in the previous submissions. Otherwise, the papers will be immediately rejected. They simply don't fit into a pharmacological journal, and we see the great interest of the community, but we have to make sure that we look on mechanisms.
Okay.
Fully agree.
Okay, so, Valerie, thank you very much. And you need...
Okay, now we will move on, and the next presentation will be given by Dr. Wen Zhang from Beijing, from the Institute of Materia Medicalis, and the title is Salvianolic acid A, a natural product from Salvia meteoriza bunge, alleviates cerebral ischemia, reperfusion injury via the inhibition of FoxO3a beam signaling pathway. Please.
Okay, thank you for the chair's kind introduction. It's a pleasure to be here, and it's my honor to speak before you at this pharmacology conference. Today, I would like to discuss the topic of stroke and the natural protective effect of salvianolic acid A for the treatment of ischemic stroke or cerebral ischemia reperfusion injury. My title of the topic today is Inhibition of FoxO3a beam signaling pathway, and the MMP9 contributes to the protective effect of salvianolic acid A against the cerebral ischemia reperfusion injury. As we all know, stroke is a growing global burden, with being types of ischemic stroke, hemorrhagic stroke, and transient ischemic attack. Epidemiological studies have revealed that there are over 1 million stroke-related deaths worldwide each year, and ischemic stroke accounts for approximately 87% of all stroke cases. But so far, TPA is the FDA-approved thrombolytic agent for acute ischemic stroke, but with narrow therapeutic window and high risk of hemorrhagic transformation. Therefore, it is important to find the new drug to treat ischemic stroke. The cellular and the molecular mechanisms of stroke are complex. Multi-target mechanisms and cells participate in the pathological process of stroke, and the pathological stages and sequence of damaging events is complicated. Therefore, the development of multi-effects of drugs to treat ischemic stroke is become necessity. Then, in 2001, the scientists in United States proposed the concept of neurovascular unit. Nowadays, the protecting has shifted from solely protecting neurons to protecting neurovascular unit, containing the neurons, endothelial cells, microvessels, and glial cells, and some other supporting cells. As we all know, plants is a valuable source of medicines for centuries, and with nearly 200,000 natural products discovered to date. And China is one of the major discovered to natural products research. Natural medicines have been used in China to treat ischemic stroke for many years. And an analysis of 189 clinical trials involving 19,180 patients across 22 traditional formulations evidenced the effectiveness of the traditional Chinese injections in improving patients' neurological deficit. However, some drugs need further validation of their therapeutic effect and the validation of their action mechanism in order to be approved abroad. Danshen is a medical herb widely used in traditional Chinese medicine. Modern scientific research have revealed that danshen has a variety of pharmacological activities, including antioxidation, antiplatelet aggregation, protecting the cells from apoptosis, inhibiting ischemia reperfusion injury, promoting the endothelial cells, acting on lung channel functions, anti-inflammation, and so on. Salvianolic acid is an active natural product extracted from danshen. Previous mechanism studies of salvianolic acid in stroke models have indicated that salvianolic acid could downregulate RS, inhibit soluble epoxide hydrolyze. Other disease models also indicated that salvianolic acid has a variety could regulate a variety signaling pathway, including activate of AMPK PGC1 alpha SIRT3 pathway, and downregulate the phosphorylation of JNK signaling pathway, upregulate the phosphorylation of ERK, and activate the BMPR2 and the SMART signaling pathway, activate NERV2, upregulate the anoles, inhibit the AGE and the RAGE signaling pathway, and so on. So in this study, we will first confirm that salvianolic acid could the protective effect of salvianolic acid on blood-brain barrier and the neurovascular unit by inhibit MMP9, and then we will confirm that salvianolic acid could inhibit cell apoptosis by promoting the phosphorylation of FoxO3a. So first, in vivo study, we used the right MCL reperfusion injury to elevate the salvianolic acid the therapeutic effect. The result showed that MCAO reperfusion injury could increase the right neurological deficits and significantly increase the right brain water content. Well, salvianolic acid A treatment could reduce the neurological deficits and reduce the brain water content in a concentration-dependent manner. TTC staining was used to assess the right brain infarct volumes. So MCAO reperfusion injury will significantly increase the infarct volumes. Well, salvianolic acid A treatment could significantly reduce the infarct volumes in a concentration-dependent manner. Then, HE and nasal staining show that salvianolic acid A could elevate the brain injury in IR rats. In addition, salvianolic acid A could inhibit the oxidative stress, downregulated the MDA production, upregulated the SOD and GPX levels. For further study, we focused the protective effect of blood-brain barrier. As shown in Figure A, MCO reperfusion injury would increase the Evans blue elucidation and severe result in tight junction proteins degradation, and the salvianolic acid A treatment could inhibit the Evans blue elucidation and inhibit the tight junction proteins aggregation, including the protein levels of CL1, claudin 5, and aclaudin. For further Western blotting, studies have revealed that MCO reperfusion injury would increase the protein levels of MMP9 and MMP2 and decrease the protein levels of TIMP1. Well, salvianolic acid A treatment could inhibit the IR injury-induced MMP9 upregulation and inhibited the TMMP1 downregulation. Then the MMP2 and MMP9 expression were detected by the glytin-thermography assay, as shown in Figure D. Salvianolic acid A treatment could significantly downregulate the expression levels of MMP9, but has no significant effect on the MMP2 protein levels. So our in vivo study verified the therapeutic effect of salvianolic acid A. The further study, we will focus on the action mechanism of salvianolic acid A in vivo and in vitro study. First, in vitro study, we use the OGDR stimulation. The OGDR stimulation will result severe cell damage and cell viability loss. Well, salvianolic acid A treatment could inhibit OGDR-induced cell viability loss, as shown in MTT assay and under the inverted microscope in a concentration-dependent manner. Well, in vitro study, the 5 micromole concentrations of salvianolic acid A showed the maximal protective effect of cell viability loss, so it was selected for further study. As we all know, FoxO3a regulates the cell apoptosis, and OGDR stimulation downregulated the phosphorylation of FoxO3a and AKT. Well, salvianolic acid A treatment could upregulate the phosphorylation of AKT and FoxO3a. As shown in Figure C and Figure D, OGDR stimulation also downregulated the phosphorylation of mTOR and GSK3 beta. But salvianolic acid A treatment could not have no significant effect on the protein levels of mTOR and JSK3 beta. So further study was focused on the regulation of FoxO3a by salvianolic acid A. In addition, OGDR injury also upregulated the protein levels of BIM and cleaved caspase-3, which represents the cell apoptosis. While salvianolic acid A treatment could downregulate the protein levels of BIM and cleaved caspase-3 in a concentration-dependent manner. The above results indicate that PI3 AKT and the FoxO3a signaling pathway contributed to the neuroprotective effect of salvianolic acid A on cell apoptosis. So to find out whether the phosphorylation effect of FoxO3a and the AKT the 5 micromole concentrations of salvianolic acid A were added and incubated for 1, 2, 4, and 6 hours. And different concentrations of salvianolic acid A were pretreated for 4 hours. The results indicate that salvianolic acid A could increase the AKT and FoxO3a phosphorylation in a time and concentration-dependent manner. So the above results showed salvianolic acid A could increase the phosphorylation of FoxO3a and AKT. Then to find out whether the PI3K AKT signaling pathway could contributed to salvianolic acid A-induced FoxO3a phosphorylation. The PI3K inhibitor was used. And firstly, the cells were pretreated with the PI3K inhibitor for 40 minutes before the cells were subjected to OGDR stimulation and the salvianolic acid A treatment. Then the MTT assay was carried out. As shown in Figure A, the pre-incubation of the PI3K inhibitor could reverse the salvianolic acid A-induced cell neurological effects. And as shown in Figure B and Figure C, the pre-incubation of the inhibitor would reverse the salvianolic acid A-induced the phosphorylation of AKT as well as FoxO3a phosphorylation. As shown in Figure E, salvianolic acid A treatment could inhibit the nuclear translocation of FoxO3a, which was also reversed by the interference of PI3K inhibitor. Then, to evidence the FoxO3a beam signaling by interference the FoxO3a, the FoxO3a siRNA were introduced. As shown in Figure E, OGDR injury could upregulate the nuclear FoxO3a levels, while salvianolic acid A treatment could downregulate the nuclear FoxO3a levels, which was partly reversed by FoxO3a siRNA, which triggered the downregulation of protein levels. So these results show that PI3K AKT mainly contributed to the salvianolic acid A-induced phosphorylation of FoxO3a in SY5Y cells, and the phosphorylation of FoxO3a would downregulate the nuclear FoxO3a levels, which further downregulated the protein levels of BIM. The above in vitro study, we studied the action mechanism of salvianolic acid A, that salvianolic acid A could inhibit cell apoptosis via PI3K AKT and FoxO3a signaling pathway, Well, if salvianolic acid I had the same effects and mechanisms in vivo study, we also used the right MCL reperfusion models. As shown in Figure A and Figure D, the MCL reperfusion injury could downregulate the phosphorylation of FoxO3a and AKT in the right brain cortex region, and the salvianolic acid A treatment could inhibit the downregulate of FoxO3a and AKT phosphorylation. In addition, the upregulate phosphorylation of FoxO3a and AKT was reversed by the interference of PI3K inhibitor. And as shown in Figure D, MCAO injury also downregulated the phosphorylation levels of FoxO3 and AKT in right brain hippocampus region, and salvianolic acid A also could increase the phosphorylation levels of AKT and FoxO3a, which was also reversed by the inhibitor treatment. Then, in immunofluorescence assays showed that salvianolic acid A could inhibit the nuclear translocation of FoxO3a in neurons, both in right cortex and hippocampus region, which was also partly reversed by the inhibitor of PI3K treatment. In addition, MCL reperfusion injury also upregulated the expression of BEAM and collude caspase 3, which represents the cell apoptosis in right cortex and hippocampus regions. And the salvianolic treatment could significantly inhibit the expression levels of BEAM and collude caspase 3 in a concentration-dependent manner. So the TL staining was assessed the cell apoptosis rate, as shown in Figure E and as shown in Figure A and Figure C. MCL reperfusion injury model could significantly increase the cell apoptosis in neurons, both in cortex and regions, and the salvianolic acid A treatment could significantly inhibit the neurons cell apoptosis rate, which was reversed by the PI3K inhibitor. And as shown in Figure E, MCL reperfusion injury could downregulate the cell, could inhibit the cell apoptosis of GLUT1 positive cells. Well, salvianolic acid A treatment could inhibit the percentage of GLUT1 positive cells apoptosis, which was also reversed by the PI3K inhibitor treatment. And the TTC staining was used to assess the right brain infect volumes. As shown in Figure J, there was almost no infect volumes in sham group, and MCL reperfusion injury result in severe infect volumes compared with the sham group. Well, salvianolic acid A treatment could significantly downregulate the percentage of right brain infections, and the treatment of PI3K inhibitor would partly reverse the salvianolic acid A effect of the brain infections. Well, MCL reperfusion injury also result in the neurological impairment with the same group, while salvianolic acid A treatment could downregulate the right neurological deficits, which was also could partly reversed by the PI3K inhibitor. So in our in vivo and in vitro studies, we also verified the therapeutic effect of salvianolic acid A and the action mechanism of salvianolic acid A in MCL reperfusion injury and OGDR reperfusion injury. In conclusion, salvianolic acid A could elevate the cerebral ischemia reperfusion injury at least partly via the AKT FoxO3a and beam signaling pathway, and protecting effective on blood-brain barrier and neurovascular effect by inhibit the MMP9. This is all my report. Thank you for your attention.
Thank you very much for this beautiful study. This paper is now open for discussion. Yes, please. I don't think the microphone is working. Closer.
Do you have any data?
No, not working.
Sorry, I can't hear.
Just need to speak close to the mic, please. It is on.
Do you have any data to support that this compound work as neurogenesis also? Any data to support neurogenesis in a stroke model?
Is your compound working on neurogenesis, is the question.
The salvianolic acid is an active natural product from dantian, and another natural product, and dantian has also another active natural product like salvianolic acid A, B, C, D, E, F, and so on. But salvianolic acid A is discovered by my supervisors and have studied for many years. And we have the confidence that salvianolic acid A is a successful new drug for treat the ischemic stroke. And we use the middle cerebral artery occlusion and the oxygen growth OGDR stimulation, these models to find out whether salvianolic acid could have the therapeutic effect.
Thank you. Any further questions? Very short question, short answer.
Yeah, just a quick question. I will read from here. So what is known about the bioavailability of salvianolic acid? Because I guess dantian is used orally in China. So any clue about bioavailability of this type of compound? If it gets metabolized, or because in your assay you show very nice in vivo data, I guess the compound was even IV.
Yeah, the traditional dantian is a medical herb widely used to treat ischemic stroke for in China for many years, and and has always made the traditional Chinese injections. And start.
Done by injection.
Yeah.
Thank you very much. We now have to move on because time flies. Thank you, and I would like to introduce the next speaker, which is Dr. Xiaohua Lu. She comes from the University of Mainz in Germany, and she's part of the team of Thomas Efferth, and her title is Network Pharmacology of Angiogenesis Inhibitors Inspired by Nature. Please.
Okay. Hello, everyone. First of all, I would like to express my appreciation for the kind invitation and also introduction. So it's really a great honor and pleasure for me to share our works. Today, the topic will be network pharmacology of angiogenesis inhibitors inspired by nature. So what is the network pharmacology? In the recent years, the establishment of the series of biologic database have been developed for the to support the pharmacology research for a certain disease. The database such as Schini-Kerth or OMI could be used to identify what kind of genes could be used to could be identified to figure out what kind of genes could contribute to the disease. For a specific compound, the database such as the Swiss target prediction and PharmaMapper could be used to figure out what kind of genes could be affected by this compound. In this way, a set of common genes could be easily obtained to construct the disease's phenotype gene target drug interaction network. The following database could be used, such as the strain cytoscape and the ingenuity pathway analysis, as well as the in silico techniques. Then further studies in vitro, both in vitro and in vivo, could be used to validate the effect of this specific compound on the diseases network. Cancer is the second leading cause of death worldwide after cardiovascular diseases. It's estimated 19.3 million new cancer cases and around 10 million cancer cases in 2020. A huge amount of the money has been used to support for the cancer treatment. With the development of the medical research, a series of anti-cancer strategies have been developed. Even though cancer is still the most stubborn disease in the world. So you may ask, what are the causes of cancer? Why they are so difficult to overcome? Generally speaking, cancer is a genetic disease. It is caused by the genes that control the way our cells grow, divide, and function. There are three types of genes: oncogenes, tumor suppressor genes, and the DNA repair genes. The changes of these genes could help the cancer cell to grow out of control, to invade the growth suppressors, and to activate the invasion and metastasis, as well as to induce the angiogenesis. Inhibitors of this changed genes or the proteins could be the promising anti-cancer agents. As we can see here, inducing angiogenesis is one of the main hallmarks of cancer. Inhibitors of VEGF signaling pathways could be the promising anti-angiogenesis strategy. So what is the angiogenesis? As the tumor grows, it eventually reaches a size where it requires additional vasculature to sustain continued growth. To reach this goal, the tumor cells secrete certain proteins to promote the blood vessel growth around and into the tumors. This process is called angiogenesis. Meanwhile, the cancer cell can transfer to other sites of the human body through the blood vessel to form the metastasis tumors. Now let's take a closer insight into the VEGF and their receptors. VEGF are the main drivers of angiogenesis. This family includes 5 members: VEGF A, B, C, D, and placental growth factors. VEGF bind to their receptors, leading to its phosphorylation and also the formation of the homodimers and heterodimers. VEGFR includes 3 parts: extracellular domain, transmembrane domain, and kinase domain. VEGFR1 mainly responsible for the development of hematopoietic cells and the migration of the monocytes and the macrophages. VEGFR2 mainly responsible for the angiogenesis, migration, proliferation, and survival of endothelial cells. VEGFR3 mainly responsible for the development of the lymphoendothelial cells. So VEGFR2 play a dominant role in the angiogenesis. In the market, we can easily find the anti-VEGF or the anti-VEGFR2 inhibitors. However, they display the side effects such as the hypotension, thyroid dysfunction, and cardiotoxicity. Therefore, it's necessary to look for the novel anti-angiogenesis inhibitors. Lateral products have historically played a key role in the drug discovery, especially for the cancer and infection diseases. In the latest 40 years, almost 50% of the FDA-approved drugs are derived from the lateral products. Recently, we wrote a review which summarized the anti-angiogenesis lateral products. In this review, we summarized around 150 lateral products. The underlying mechanism for these lateral products is that they could inhibit the VEGFR2 signaling pathways, thus to inhibit the vascular permeability, cell migration, and the cell proliferation. In our lab, we are specifically interested in the artemisinin type compounds. As we all know, for this type compounds, they have been developed as the standard therapy for the malaria treatment, while the biologic activities for the artemisinin type compounds are not limited to the malaria treatment. They also, this type compounds, they could also have the effects on various diseases. Such as the viral infection, schistosomiasis, trypanosomiasis, and so forth. The most important part is that the artemisinin type compounds could inhibit the cancer. Not only in our lab, but also in other labs, we proved that the artemisinin type compounds inhibit the cancer through multiple ways. It could arrest the cell cycle, and it could also to induce the apoptosis and to inhibit the angiogenesis. So our question is there any artemisinin type compounds which could target VEGFR2, therefore to inhibit the angiogenesis? This is our project goal. As an initial step, we performed the clinical relevance to figure out the prognostic potential of VEGFR2. In the second step, we performed the molecular docking to look for the most potential artemisinin type compounds, which has the favorable binding affinity with VEGFR2. In the last step, we performed both in vitro and in vivo studies to validate the anti-cancer mechanism of the selected artemisinin type compounds. And now let's come to the results part. Here is the prognostic potential of VEGFR2 in different cancers. We applied the ProgloScan to check the survival rate of the cancer patients with high or lower expression of VEGFR2. The Kaplan-Meier plot indicates that the higher expression of VEGFR2 in breast cancer patients is correlated to a lower survival rate. This is also observed in colorectal cancer, esophagus cancer, eye cancer, lung cancer, as well as the overall cancer. In the following slides, I would like to introduce two artemisinin type compounds, which were reported in our group last year. These two compounds has showed a favorable anti-angiogenesis effect. The first one is the FoES123. How this compound was selected out? We obtained 52 and 364 artemisinin type compounds from the PubChem and PubMed respectively. In the multi-docking, here is the bar chart for the number of the artemisinin type compounds in response to different category of the binding energy with VEGFR1 or 2. The reference compound artesunate showed a binding energy at around minus 9.7 kilocalorie per mole. 194 and 245 artemisinin type compounds showed a lower binding energy with VEGFR1 or 2 in comparison with the artesunate. It means that most of the artemisinin type compounds had a good binding affinity with VEGFR1 or 2. This is the linear correlation between the noticed binding energy and the predicted inhibition constant. It means that this molecular docking method is well established. In the most interesting point in this part is that the top 10 artemisinin type compounds bind to the similar space of VEGFR2. 4 of them are commercially available, so they are used for the further experiments. FoES123 was finally selected out because of its strong cytotoxicity on different cancer cells. Here, the breast cancer cell line, MDAMB231, is the most sensitive solid tumor cells with the IC50 value around 10 micromolar. The leukemia cells, CCRFCM and CEM-ADR5000, showed even higher sensitivity toward this compound. The IC50 value for the peripheral blood mononuclear cells was around 84 micromolar, which means that this compound had a meaningful effect on the human healthy cells. When we look back to our molecular docking result, we found that FoES123 could be stably located at the ATP binding pocket of VEGFR2. A list of the 18 residues are responsible for this effect. Human umbilical vein endothelial cells, Huyck cells in short, is a commonly used cell model to study the angiogenesis function of the endothelial cells. Here, this compound could inhibit the cell viability of Huyck cells at around 4 micromolar. In the presence of VEGF, the cell viability of Huyck cells is increased around 15%. FoES123 had less effect on the cell viability of the VEGF-induced Huyck cells. Here is the tube formation result. VEGF could enrich the formation of the capillary-like tubes, while FoES123 could disrupt the tube formation, as evidenced by the sharp decrease of the branching point and the turbulence. Cell migration is an initial step for the cell invasion. Both the cell migration and the cell invasion are dependent on the tumor angiogenesis. Here, we performed the 2D wood AC and the 3D spheroid AC on the most sensitive solid tumor cells, MDAMB231 toward FoES123. As we can see here, FOS123 could inhibit the cell migration on these both models in a time and dose-dependent manner. Likewise, FOS123 could suppress the cell invasion in a dose-dependent manner. Here is the apoptosis. Usually, cell apoptosis is carefully regulated in the human healthy cells, while for the cancer cells, due to the genetic changes, the cancer cell can not only grow out of control, but also to lose function of the apoptosis. Here, we performed the cell apoptosis using the flow cytometry. This diagram shows the different cell population. Once the cancer cells undergo the apoptosis, the cell population would shift from the right to the left and the top. As we can see here in the bar chart, the cell apoptosis was significantly induced by this compound FOAS123. To have a real interaction between FOS123 and VEGFR2, we performed the microscale thermophoresis experiments. As we can see here, this curve, it means that FOS123 could strongly bind to the VEGFR2 with the dissociation value at around 1.3 micromolar. To further check the relevant signaling pathways, we also performed the Western blot. We observed that not only the phosphorylated VEGFR2, but also the phosphorylated GNK, ERK, and AKT were reduced. Chloreal anatoic membrane assay, CHEM assay in short, is a commonly model to screen out the angiogenesis agent in the pharmaceutical research. This method is quite easy, inexpensive, but highly reproducible. So in this experiment, FOS123 inhibited the formation of the blood vessel, and also to inhibit the formation of the branching points. That is all the results regarding our first compound, FOS123. The second compound is FO8643. Yes. For this compound, since we applied the similar techniques and methods, I will not go into so much detail. Here, I just give you an overview or a summary. For FOS643, we also tested the cytotoxicity on different cancer cell lines. The liver cancer cell line HUH7 was the most sensitive solid tumor cells. This compound could also arrest the cell cycle and the G2M phase. This FOS643 could also induce substantial apoptosis in the microarray assay and the angiogenesis assay. This compound could also inhibit the cell migration and also the angiogenesis. That's all the preliminary experiments we have done so far. But their story about these two compounds will not stop here. For the next step, we are also planning to study the following perspectives. So we would also like to investigate the correlation between cancer stem cells and angiogenesis to check the metastasis mechanism and the anti-tumor activity as well as the side effects. Okay, so I am really thankful to have the CSC grant to join Proof Effort group. And thank you for your attention. Now I'm open to your questions.
Thank you very much for this very nice presentation, which is now open for questions. Yes, please.
Yes, this is a wonderful work you did in the Efferth lab. And Thomas Efferth is one of the editors dedicated to the natural compounds. And this is a wonderful study where you do network pharmacology not only in silico, but with real pharmacological experiments, because there is now an inflation of submissions just with a virtual docking, and this is not really useful for. You really need experimental validation, so congratulations to this wonderful work.
Yeah, thank you for your for a question.
Yes, yours.
Geneva, just the second the compound that you mentioned briefly, what is the IC50? Because the first one was 1.3 micromolar, which is okay, but not great. And how much is the second one?
For the second one, actually, we also, because of the time limitation, I didn't put so much detail. For the second compound, F08643, we also performed the resuscitation-based cytotoxicity assay on different cancer cell lines, like the leukemia cells and the solid tumor cells. For the solid tumor cells, it's around 10 to 20 micromolar. Yes, and for the suspension cell line, such as the CCRF or CCRF CEM or CEM ADR5000, it's even much more lower. It's around, if I remember correctly, it's around 0.3 micromolar. If you need more details, you can also refer to our publications. Yeah.
Okay.
Yeah. Thank you.
Thank you.
Yes, please.
I think I appreciate your presentation and the data. I have a couple of queries. Actually, we have done studies in 2006, artemisinin compound in experimental induced colon cancer, and it was found to be very, very efficacious. So when we carry on that work, we had only challenges because this is a chronic therapy. You have to give it for a long period of time. And challenges it comes that it also affect the brain, artemisinin compound. So how do you address the issue tomorrow when you go to clinics that it is going to affect the brain in terms of seizure-inducing agent? So how do you address in terms of the dose selection and effect on the CNS? Thank you.
Okay. Thank you for your question. Actually, this is really a good question regarding the artemisinin type compounds dose selection. Even though we didn't, we don't perform any dose information on the in vivo studies so far. As you can see in our presentation, we only performed the Kim-AC. But as you know, for the in vivo studies, like the rats or the monkeys, this would be the preliminary in vivo studies. And like when we talk about the clinical studies, I think we need to put more effort on this. Yeah. I hope I answer your question. Yeah.
One last question, please.
Very nice presentation. My question is almost same. What's the status of artemisinin in other types of carcinomas apart from breast cancer in which we have studied?
Sorry, I didn't really get your point. Could you please?
Can we use this artemisinin derivatives in other types of cancer? We have studies on breast carcinoma, na?
If I understand you correctly, you mean if this this artemisinin type compounds can be applied to the breast cancer?
Other types of other cancers, other carcinomas, can we?
Yeah, based on our first experiments, the resuscitation-based cytotoxicity. For this type compounds, they do have different. They do have the cytotoxicity on different cancer cell line. So for the breast cancer cell line, MDMB231 was the most sensitive one. I believe, like, since we just use a limited number of the cancer cell line, even though this cell line from different cell type, but I think we need to test more cancer cell line to check the possibility or the potential of this artemisinin type compounds and the cancer.
Right. Thank you.
Thank you too.
Thank you very much. No more questions? Then thank you, Dr. Lu, and thank you. And we now will conclude the session. I would first of all thank all the speakers for a wonderful presentation. I think we are convinced that from the phytochemistry level up to preclinical and clinical data, there is potential for natural products, but we need very good characterization, identify the active pharmacophore, and of course also look at the end for toxicity. And thank you very much for the audience for contributing, and I give the last word to Professor Spedding.
I think it's been actually a great day for me as well, because I started to look for, as Secretary General of UFAR in 2014 to advance the field, and I think you can see just how far it's gone. And it is getting quite a long way. There's an issue I'd like to bring up for the editors, that Dr. Wolfender has clearly shown a way forward to ensure we can see which compounds are going to be active and which compounds are going to be toxic, and whether these compounds are acting in synergy or not. So one of the main reasons I wanted to start this was to prevent people just putting mixtures into animals and it not going any further. And I think now we're at a stage whereby we can begin to bring it together so we can see groups of compounds now. I think that's scientifically valid to be published. So I think that's a point I would like to make. And I think I would just like to thank everybody again, and my co-chair and worked together for ages, Valerie. And so thank you for a great seminar.
Thank you. We close the session. Thank you.